All right. Hello, all. Welcome to Lab 4. Remember, this is not on the quiz that is going to be this week, but this is your lab for the week. So, again, this is for both classes. And we're going to be talking about mendelian and monohybrid genetics. So, we'll explain what those words mean. One of the things I wanted to address here is that the other classes that we've taken, especially Lab 2 with the analysis, that relates very directly to here because a lot of Mendelian genetics is about ratios. And you can use chi-squared to address and test whether or not a certain ratio that you expect is actually happening. And this can also tell you whether or not you're having a specific inheritance pattern. So we're going to start and talk a little bit more about exactly how genetics works, and then we're going to go back to how this was discovered. Okay? All right, so basic knowledge. Some of you guys know this. There is a company called 23andMe that tests your chromosomes and such. So that's because you have 46 chromosomes. You have 23 pairs. So you have 23 from your mom and 23 from your dad. Each chromosome has genes. So you have up to 20,000 of them across your entire genome. Remember that genes go through the central dogma. They are read off of the DNA to RNA and turn into proteins. And those proteins make everything you are. I have genes for curly hair. I have genes for pale skin and freckles. I have genes that make me more susceptible to Crohn's disease. Actually, I have a genetic marker that they were able to test for. So this is the key here, is that these genes help us make us who we are. And we're going to talk a lot about genes in the next several weeks, especially as we get into evolutionary processes and understanding how genes are acted on by evolution. So again, when genes express, they produce proteins. They eventually end up creating all the traits that you have, even some personality traits. Personality is a little bit complicated though. So how does this happen? Well, we have talked before about reproduction and growth. These are characteristics of living things. Sorry, pardon me. Drink. So the ability of organisms to produce more of their kind distinguishes them from non-living objects. Its continuity of life is based specifically on reproduction of cells, which includes cell replication and also creation of gametes. Those gametes will be able to reproduce new organisms that look like their parents, and also basically the continuity of life, creating a, so having a baby that goes to a child, that goes to a teenager, an adult, and then continues with the reproduction of new life. So the goal of most cell division is that you create two identical daughter cells. So you want to replicate the cell in its entirety. And we're not going to go through all the stages of mitosis and meiosis. This is, again, not cell biology, just understanding, though, that this, when this happens, all that happens in your cells is you line up all the chromosomes that have been duplicated and you split them apart. Okay, and we're going to talk a little bit about how the chromosomes work. The DNA is copied and organized exactly. You want a genetically identical copy of the original. This means that you go skin cell and have cell division and you still have a skin cell. This gives you an exact replica. So every cell in your entire body, besides the sperm and eggs, contains 23 pairs of chromosomes. These two chromosomes in each pair are called homologs or homologous chromosomes, okay? They are two separate chromosomes. They are not attached to each other. And they can carry different versions of genes, okay? They're the same length and shape. They carry genes controlling the same inherited characters. So if your mom has brown eyes and your dad has blue eyes, then you have the homologs that carry those genes will have one blue allele and one brown allele. Chromosomes are these organized packets of DNA. Okay, so the idea is that it's kind of like having your socks. In order to make sure that you, if you wanted to say, put half your socks into a suitcase, if you just reached in and grabbed a bunch of loose socks, you'd probably get a lot of mismatches. And we don't want that, especially for replicating cells. So DNA is packaged in the chromosomes so the pairs stay together. And not the homologs in particular, but the identical pairs. And so we're talking about identical pairs. So these are organized packets of DNA, and they're woven together with protein strands called chromatin. And they exist in the nucleus. They coil up during cell division, and this facilitates proper division. Okay? So this is a duplicated chromosome. Now I'm going to link a video here. I'm not going to show it right now, but I will show you where you can find it. Next explains a little bit about what a duplicated chromosome is, which is this, okay? So this is one chromosome and two chromatids are stuck together, okay? So what it means to count chromosomes when they're duplicated, and how homologs work, okay? So I will link that. It's from a great little educational series on YouTube called Amoeba Sister. They use them all the time. So these pairs of homologous chromosomes, okay, when they're getting ready for division, these guys in a meiotic division, they're going to line up one by one, okay? And each of these sister chromatids on either side are, the sister chromatids are identical to each other. So if you line them all up and then you divide them equally, you have identical on both sides, okay? But, so this is a picture of the actually these might be duplicated, I can't actually tell from here. But a cell with two sets is called diploid. We will talk about other ploides, that's what they're called. But for humans, we are all diploid. There cannot be a triploid or a quadruploid human. There can be for plants. Humans cannot survive multiple ploides, okay? We have to have two copies. That's just how we're built. So this is called 2n equals 46, which means n is 23, hence the 23 pairs of chromosomes. So this gives you a little bit of an idea of how this works, okay? So let's pretend you have a diploid cell. It only has 6 chromosomes, which means the n number is 3. Maternal set of chromosomes, n equals 3. Paternal set of chromosomes, n equals 3, okay? 3 from your mom, 3 from your dad. So you have a replicated chromosome here, or a duplicated chromosome, okay? So these two are two sister chromatids. They are identical to each other. They're on one replicated chromosome, okay? And if you have two of the, if you have the homologous pair together, these are two non-sister chromatids. And we're going to look at something special that those non-sister chromatids do in, these non-sister chromatids do in meiosis, which is a different form of division. The centromere holds these together, and we call them one chromosome until the sister chromatids divide. Right now, though, you wouldn't call it, we wouldn't say that we had 12 chromosomes, even though technically it is. Remember that they all line up with their centromeres aligned in one line and divide equally their sister chromatids for mitosis, the identical division. But there's a different kind of division that's important for understanding how gametes are formed and how inheritance works, okay? So you have this homologous pair of chromosomes and a diploid parent cell, okay? This is going to be a germline cell, so something that's in your ovaries or your testes, okay? Then this duplicates. Now you have the duplicated chromosome, okay? And you have these sister chromatids on one, and then they align together with non-sister chromatids next to each other. So they align in pairs. And then one of each pair moves to either side. The sister chromatids are not divided right now. There's actually two divisions that happen. Once these two cells have formed, it immediately goes through a new division. Now, we started with a diploid, and we go to what's called a haploid, one copy of each. Two of them will have this chromosome with your dad's, and two of them will have this chromosome with your mom's, okay? It's different than mitosis. This creates non-identical and cuts the number of chromosomes in half, which is important because if you want to have one haploid and one haploid come together for a diploid, because we don't want to be a quadruploid human. The first division separates the homologous chromosomes, so that's this one, okay? You still have the duplicated chromosome, but now they're no longer together. Remember, in mitosis, if you're just duplicating your body cells, that's in order to duplicating your body cells is for creating identical cells. The goal is to create haploid, only egg in the sperm. Second separates the sister chromatids. So you get 4 from each because there's a total of 4 chromatids. The goals of meiosis are to go from diploid to haploid, and this prepares it for fertilization, okay? The goal is also to create genetically different cells. And you say, well, hold on, these two are identical and these two are identical. You think that, but we're going to walk through another activity that happens during meiosis that makes it so that they will be non-identical. In meiosis 1, when they are lined up together, these two non-sister chromatids cross over, and they share a little bit of information. So you got a little bit of your moms and your dads and a little bit of dads and your moms, okay? When they get pulled apart, one of their chromatids now looks different than the other one. So now when we pull them apart, you have one fully dad, one fully mom, and then 2 recombinant. We're only looking at one division of one chromosome pair. Remember, you have 23 of these. And so that happens with all of them. And so you are made of a combo of your parents, and possibly some of them are fully dad. Some of them are fully mom. And so you may be fully mom for one chromosome, fully dad for another chromosome, and they have recombinant ones for a variety, okay? All right. And there's something else called independent assortment. We're going to work through independent assortment as it is, but remember, every time you line up in meiosis 1, they're lining up in pairs. But imagine each one of these lines as a coin flip. You can have mom one side and dad the other. And they can be lined up all the way down like this for 2 combinations. But if they're flipped, which can happen to any of them, then now you have your dads go over here and your moms go over here. 3 and 4. But now multiply that by 23. 23 squared is the number of flips that can happen, number of combos you can create via this kind of flipping. Independent assortment means that the orientation of these 2 chromosomes is not related to the orientation of these 2 chromosomes. That's going to matter when next week we get into more complex 2-gene relationships, okay? And it has to do with that and/or rule. We're going to talk about that as we move forward here, but this is one important thing to know is that chromosomes orient independently of each other, which means that your eye color gene is not related to, let's say, whether or not you have a widow's peak. I was going to say your hair color, but it is actually related because that's all melanin related. So eye color is not related to something like your height, okay? Those are independent variables. Like flipping a coin twice. The second flip of the coin does not rely on the first flip. Similarly, parents with 2 children, just because you've had a boy doesn't mean you're likely to have a girl. It doesn't balance like that because it is that 50-50 chance. And that has to do with the Lab 2 statistics that we took. All right, so we have 2 different ways that, that duplication can happen. Sexual reproduction, meiosis, going from diploid to haploid gametes, and asexual reproduction, mitosis. All right, next we're going to move into actual inheritance and how the segregation and dominance and independent assortment affects inheritance patterns, okay? So we have to understand what principles account for the passing of traits from parents to offspring. Originally, there was this blending hypothesis, the idea that genetic materials from 2 parents blend together. Blue and yellow paint make green. And this likely came from observations of parents of different hair color or skin color. They tended to have children with halfway in between. So that's what's called a polygenic trait. We're not going into that today, but most traits are not polygenic in that you don't have one gene contributing a tiny bit here and there. Instead, it's what's called particulate. Parents pass on discrete heritable units. Now, technically, the discrete heritable units, technically, those actually do so those discrete heritable units actually do influence that blending of skin tones because each individual packet contributes a little bit here and there. And we'll talk about that later. But the particulate hypothesis is how this actually works. So Mendel documented a particulate mechanism through his experiments with garden peas. So who was Mendel? He's the pioneer of the study of heredity. When we talk about Mendelian genetics, we're talking about the basic Mendelian principles that he created through his meticulous research. He was a monk, a religious scholar. He had a garden full of peas, and he crossed these garden peas, analyzed their offspring, and derived important concepts. And we're going to go through some of these concepts. But the segregation of 2 parental forms of a gene, he was the one who figured out that you get 1 copy from your mom and 1 copy from your dad. He didn't know what these copies were. There was no information. This is a long time ago, before the discovery of DNA or chromosomes. Okay, he actually discovered this. So it was shortly after. This was shortly after Darwin went on his trip. So they didn't really correspond with each other. He also identified the independent assortment of 2 or more parental traits among offspring. So why did he use peas? They were available to Mendel in many different varieties. He could get different colors, dwarf plants, tall plants. He could get varying colors of pea pods, and he wanted to understand that because he knew he could get a yellow pea pod. He knew he could get a green pea pod, but there were no yellow-green pea pods. He wanted to understand why. Additionally, he could get purple peas, and he could get white peas, but he could get no violet peas. Why is that? Was he never able to find violet peas? He used these because they had a short generation time. He could create large numbers. Remember, large numbers important for statistical analysis. and he could control the mating exactly. He could make it so that they would be allowed to self-pollinate or cross-pollinate. So I'm going to show you how he did that. In the parental generation, he would, if he wanted to do a cross-pollination, before the, these are called the stamens, okay? Before the stamens matured, he would cut them off. Because peas can easily self-fertilize, they have both male and female parts. So he cut off the male parts before they were able to fertilize the female part. Then he would take a paintbrush and transfer from one of them to the other and put it, basically he's doing artificial insemination of plants. And then he would breed them and take the seeds from that generation and plant them and see what he found, okay? He chose to only track those characters that he knew only occurred in 2 distinct forms. He didn't understand polygenic traits, but his actual principles applied. He used varieties that were what's called true breeding. Basically, true breeding means that if you, this is the true breeding purple. If you bred this purple with itself, it would never, ever, ever, ever create a white. And we're going to explain why that is. Same with the white. You would never, ever, ever create a purple if you only bred the white with itself. In a typical experiment, he would make 2 contrasting true breeding varieties. This is called hybridization. We still do it today. The true breeding parents are the P generation. The hybrid offspring of the P generation are called F1. And then he would let the F1 individuals self-pollinate or pollinate with each other to create the F2 generation. But he made sure that only these F1s would be able to share pollen with each other. This is important because he wants to be sure of who is mating with who. Other plants, it's actually harder to control that, but with peas, because they have such a closed area and he was not letting pollinators in, it was reasonably easy to control for this. All right. So the law of segregation, some of it was that he took this pea generation. When he bred them, no matter how many offspring he created, his hybrid would be purple and not white ever. Not even light purple. And this was confusing. He did this over and over and over, and he always came with the same result. F1s, when crossed with a purple and a white, only created purple. When Mendel crossed the F1 hybrid, many of the F2 plants had purple flowers, but some had white. But when he analyzed the data, remember, data analysis is important, he always found the same ratio, 3 to 1. That 3 to 1 ratio makes sense in actually how this works. So I'm going to explain these letters in a minute, okay? So here's what he would find. First iteration, he would cross these, take the peas from the pods, and in the first iteration, would grow only purple flowers. He would let these self-pollinate, and their offspring were always 3/4 purple and 1/4 white. So what was happening here? He developed a hypothesis to explain the 3 to 1 inheritance pattern he observed in the F2 offspring. And 4 concepts make up this model, and this relates to what we just learned about chromosomes and how they work. First, alternative versions of the genes account for variations in inherited characters. These we now call alleles. He called them alternative versions. We know them as alleles. I mentioned them before. If you have a brown-eyed parent and a blue-eyed parent, then you may have one copy of the brown and one copy of the blue, and one is from each parent. But know also that then we're going to talk about the dominance concept here, okay? In the gene for flower color, there's 2 versions, one for purple and the other for white. These are alleles. Each gene, now that we know what a gene is, We know it resides at a specific spot on a specific chromosome, and each chromosome may have a different version of that allele. And for each character, an organism inherits 2 alleles, one from each parent. He didn't know what chromosomes were, but it was the only way that his data made sense. Third, important, the dominance principle. If 2 alleles at a locus differ, one of them will determine the organism's appearance, and the other has no noticeable effect on appearance. These are dominant versus recessive genes. Okay? And we'll talk about why that happens. Then, fourth, the law of segregation. The 2 alleles for a heritable character, even in someone who has 2 different versions, will segregate during gamete formation and end up in different gametes. The egg or sperm gets only one of the 2 alleles that are present in the organism, no matter what. This segregation of alleles corresponds to how homologous chromosomes separate during meiosis, which we already talked about. All right, so the dominance concept, let's run through that again. For some genes, you will only need 1 copy, 1 allele to express the gene. These genes are dominant. If your dad has brown eyes and your mom has blue eyes, blue-eyed people have 2 copies of the recessive B allele, okay? And you might have brown eyes. The reason you have brown eyes is because the copy from your dad dominates and overpowers the blue eyes. For some genes, you need 2 copies to express a gene. These genes are recessive. If your dad has brown eyes and your mom has blue eyes, you might end up with blue eyes. It means your dad must have a hidden recessive blue allele that he shared with you. Your mom only has blue alleles to share, and you end up with blue. The reason is for many things like this, if you have someone who has 2 homologous chromosomes, one has the allele for purple and one the white. When in fully dominant genes, often it means that it either creates a specific protein or it doesn't, okay? When it creates it, this enzyme synthesizes the purple pigment, and it only needs that much to create enough to color the entire plant. When it creates nothing, it would create white, but only if there were 2 copies of the white. So let's look at this in a form that we can build. Here's the P generation. Appearance is purple flowers. That's called the phenotype. Genetic makeup, 2 copies of the big P. We use big P for dominant. We use big capital letters for dominant, and lowercase letters for recessive. The genetic makeup is their genotype, okay? Big P, little p, little p. When these come together, this one can only give a big P. There's nothing else it can give. This one can only give a little p. There's nothing else it can give. They come together, and they create all purple flowers. But note that the purple flowers are not the same as in a genetic makeup, only in appearance. Instead, they have a big P, little p. Okay, we have big P, we have little p. And so here's how we put it together. When this one selfs, there are 4 different combos that can happen. The eggs can give a big P. The sperm can give a big P, making a purple plant that's what's called homozygous dominant. The egg can be a big P, and the sperm can be a little p, giving a purple that is heterozygous. And we'll talk about the definitions of these. Or remember, here's the or rule in concept and practice. Or it can be that the egg gives a little p and the sperm gives a big P for another heterozygote. big P, little p. Or the little p and the little p can come together. That's why only 1/4 of them come out as white, because you need 2 copies. Okay, so for this big P, it's what's called homozygous dominant. Egg and sperm that come from this plant, remember these plants have both male and female parts. Egg and sperm that come from it can only be big P, no matter what. So if you blend this with another big P, the true breeding, get nothing. Let me make my picture smaller, sorry. There we go. Briefly, okay. Now we have, when the egg and sperm come from the big P, egg and sperm can both either be big P or little p, and sperm can be little p or big P. That's important. This gives you more options for where it can come from. So you could combine these two and these two, or these two, or these two, 4 different combos. When something is homozygous recessive, it can only give little p's. So a white plus a white will never create a purple because there are no purple alleles to give. All right. Dominant genes are represented by a capital letter, recessive by a lowercase. If you receive a dominant brown big B from your dad and a recessive little b from your mom, you will express brown. You will have brown eyes. And you will be what's called heterozygous. Hetero meaning 2 different. Okay? If you are heterosexual, it means you are attracted to the opposite sex. So there are 2 different sexes. Okay? So think of it that way. Hetero, zygous, that means 2 zygotes came together, egg and sperm, okay? Big B, little b. If you receive a recessive blue from your dad and a recessive blue from your mom, you will express blue eyes and be homozygous recessive. You can be homozygous dominant too, but in order to do that, you have to have both parents have brown eyes. You cannot have a blue-eyed parent and a brown-eyed parent and make a homozygous dominant child, because the mother would have to contribute one of those B's, one of those big B's. If she cannot, you will not be homozygous dominant. You could have brown eyes or blue eyes in that combo, but you can only have a big B if both parents are expressing the brown-eyed gene. Again, big B children only come from parents that both express the dominant phenotype. Because even if they're heterozygotes, they have to have they both must have a big B to give. If they have no big B's to give, then they can only express the dominant phenotype. Okay. Here is one way to show how this works, okay? These are sisters. You'll note one sister looks like a normal kid, and this sister has very pale hair, pale eyes. She's what's called albino. And she has albino skin and hair. She's unable to produce melanin properly. This can come from 2 parents with normal phenotype for skin, but if they're both carriers, 1/4 of the time, they will have a albino child. So this child, also their child, she is either a carrier, 50% chance she's a carrier, and 25% chance she is normal. 25% of the time, they will have a albino child, which means it's like they rolled a 4-sided die, and once it came up on one of these 3 sides, and once it came up on this, and that's just how 25% chance is a pretty high chance. All right, so let's talk about how to do Punnett squares. What I'm going to do here, this is going to be a short video. I'm going to start out with just the basic Mendelian genetics, okay? And I will go through here. We're going to work a little bit. I'm going to show you how some of these work. But I want us to work and do this together, okay? When you're creating a Punnett square, you're going to create 4 squares. On the outside of the squares, you write down what the 2 alleles that the parent can give. If you know the parent is expressing recessive, make myself a little bit bigger here so you can see my face. If you know that parent is expressing recessive, then you would for sure put down 2 lowercase letters. Remember, for the monohybrid crosses, we're talking about only 1 gene at a time. We'll work on 2 genes next week. And up here you would put, so that it doesn't matter where the mom or the dad goes, just as long as you clarify who's mom or who's dad, okay? Up here, let's see, you put the dads, you would put what the dad has to offer. You do need to know their full genotype for this. You can't just go off of their phenotype because some dominant phenotypes, there are 2 different dominant phenotype genotypes. Okay, so here's the concept. Here we have a big P, little p, crossed with a little p. Now, I actually did this one wrong, so I'm gonna go back and I'm gonna fix it, okay? So let's actually, I'll fix it in place here, okay? So you always put this down. So you can start off and just put down, there's a big P here, there's a big P here. Always list the dominant first because think of it as overshadowing the other one, okay? Always list that one first. And then there's a little p here and a little p here, okay? So these 2 are correct. But note, this one has a big P, a little p over here. I did it right on the first one. Little p, little p. But this second one should also be little p because this one goes here, this one goes here. Now you can look at each of these as a 25% chance of getting that genotype. But if you look here, in a combo with a heterozygote and a homozygous recessive, or a heterozygote and a homozygous dominant, you actually have a 50-50 chance of getting either, okay? So we have a big P, big P, little p, okay? These 2 will express the dominant phenotype. These 2 will express the recessive phenotype. Yeah? Okay. Apologies for my broken thing here. So, because this is a complex thing, I'm going to work through the monohybrid problems, and I'm going to end the recording. I'll update this one as just monohybrid Mendelian genetics concepts, okay? And then I'm going to reopen what I had there. Apparently, I'm having trouble with my PowerPoint. It's fine. It didn't like that I fixed that. It must have been it. Let's go back down. Okay, here we go. When you solve monohybrid problems, and I'm going to run through a couple of them that are in your handout. I'm not going to do all the ones that I might actually do different ones than you have in there. I want you guys to be able to practice this, okay? Please practice and then ask me if you're right. I want you to be able to do this on your own, okay? All right, if they look recessive, you always know that they have only recessive alleles because you have to have 2 copies. If you have 2 copies of a recessive allele, you can only make gametes, eggs and sperm, with that recessive allele. That's it. For instance, I think you have a recessive trait I have. So I have a smooth, this line here is smooth. It means I don't have a widow's peak. Some people have this triangle in front of their head. The triangle is dominant. I express recessive. Actually, none of my family has a widow's peak. It's not part of my family genomes. So that means all of us cannot create gametes with that widow's peak in it. That's it. I can't contribute to a child having a widow's peak. But if my husband had a widow's peak, he could pass it on because it's the dominant gene. Okay? So if they look dominant, but you know that they have a mother or father who expresses the recessive gene. If you have brown eyes, but you know that your mother has blue eyes, you know for sure your mother must have given you one of her blue alleles. But if you don't express the blue allele, it means you have one copy of the dominant. That means that they are heterozygous. So you would put, say, one recessive parent, one recessive expressing parent. That would be the little p. You know, they're the same. And your grandfather has blue eyes, and but your dad has brown, therefore he's a heterozygote. Okay, so that's how you would do that. And then you could calculate for it. Again, it's the same one that I made previously. It's going to be 50-50 of each. But this is the key here about solving the monohybrid problems, okay? I'm going to, so here is this. Again, you have this, this is the same kind of idea. Yellow is dominant and green is recessive. So you would have a dominant Y and a lowercase y. And this is the same thing, a heterozygote plus a homozygote would create 50-50. And they just did it the opposite way, but you have the same outcome. 25 plus 25 is 50. 25 plus 25 is 50. Okay. What I'm going to do right now is I'm going to pull up, I'm going to go back. I'm going to pull up your Lab 4 online assignment. I'll do one from there, okay? And we'll work through it together. This is the simple dominance ones. We'll work on the incomplete dominance and the codominance in a separate one. Okay. So here's what your Lab 4 online assignment looks like. Now, what I'm going to do is you need to understand that this is part of the actual PDF, okay? Coin toss genetics. You're going to see this in play, okay? So you're going to pretend to be two heterozygous parents having children, okay? That means that each parent has a 50-50 chance of giving a dominant or a recessive P, okay? Again, what you can say is that Google or Siri will both be able to flip a coin for you. Probably your Androids can do it, I don't know. But you can say to Siri, flip a coin. Watch. Flip a coin.
It's heads.
It's heads, she says, okay? It'll say it too. So heads is going to be big P. Okay, let's flip another coin. Flip a coin.
Heads.
Heads. So for my first one, because I got heads are both big P. That's my first offspring. Flip a coin. Remember, this is not fixed. This is just me flipping coins. Flip a coin.
It's heads.
Again, sometimes you're going to get more dominance than others. Flip a coin.
It's heads this time.
If she gives me another heads, I'm going to do it on a different program. Flip a coin.
Tails.
There you go. So she does actually work. And then so on and so forth until you have all of them, okay? Flip a coin.
Heads.
Flip a coin.
Tails.
So I'm going to show you how to do one line here, okay? So now you have big P, big P, big P, big P, big P, little p, big P, little p. You're going to show the number with each genotype. We have currently 2 and 2. See? 2 of these. 2 homozygous dominant, 2 homozygous recessive. And, but all 4 of them are purple because each one has at least one dominant. The only time they'd be white is if I had 2 tails in a row. Do this for all of the, do this for all of them, okay? I want you to go through that and do all of them. And you're going to eventually come up with this, okay? What you're expecting is, so there's 16 total offspring that you're creating here. What you're expecting is that 4 of them will be big P, big P. 8 of them will be big P, little p. And 4 of them will be little p, little p. And you're expecting 12 to 4 ratio. You may or may not get that. This is a reasonably small thing, and you may get more big P's and more little p's, and we'll talk about that later about what's happening, but this is a small population. If you did 100 of these, 1,000 of these, more like Mendel did, then you would for sure see the 3-to-1 ratio. Again, this is a coin flip activity. But remember, now we have a coin flip where heads is dominating and tails is hidden by the heads. And because of that, we have this 3-to-1 ratio instead of a 50-50 ratio. Okay? All right. So we're going to do, I'm actually going to go back here. So these are the simple, these are just super simple monohybrid crosses, okay? But I'm going to go and pick out a different one that is not actually that, and through this, hopefully we can hold on. And working through these with me is probably a wise idea, okay? All right. Gives you all the information I've already walked through. Here's these pairs of chromosomes, etc. Here's how meiosis works. Okay? All of that. The Mendelian, coin flip genetics. Okay. Now we're going to do some of these basic things. I'm going to start here. Let's see. Find one that's not on here. Okay. Huntington's, albinism. Okay. Let's do this one together, okay? So this one, let's talk about tall stems being dominant to short stems. A flower is either tall or short. So tall and short sometimes are multifaceted, but if I ask you to set up a Punnett square, you want to set up a Punnett square, then you can go to draw, insert, table, okay? you're going to want to insert a 3x3 table. Okay, this will help you, and then if you want, you can get rid of the top and the side here. Okay. All right, so this is how I'm going to set it up. You can squish it in a little bit. There we go. All right, we have a short stemmed flower and a long stemmed one, and we get both tall and short. Remember how Mendel, he had found out that if he crossed a true breeding tall and a true breeding short, then he would only get tall. If you get tall and short, what does it have to be? To get short, I'm just going to put this here. So let's say big T, little t, okay? Choose whichever ones you'd like if I don't tell you what it is, okay? But remember that it should be always the dominant trait should be what it starts with, okay? So big T. Little t, little t. If I have any short-stemmed flowers, I have to have little t, because that's the short one. But how do I get that? Well, in order to have your Punnett square actually be able to do that, I have to have a little t here and a little t here. Now I know that one side of this is two little t's, but one of them is tall. So now you have what has to have happened. He crossed a short stem and a long stem, but he crossed a heterozygote with a homozygous recessive. Okay? So likely he got 50-50. Short and long. It doesn't say that, but that's what he should have gotten, because that's the only way you can have a tall plus a short that equals tall and short together. Okay, there's no other combos. Big T, and a little t, it would all be tall. Therefore, you know that if they create any short, you have to have that. Okay. Again, this was where we worked back from the offspring and talked about what the actual parents had to be. Okay. I'll do one from here, from your online assignment. So let's talk about this one. Blue diaper syndrome, a weird one, right? It's an unusual but harmless autosomal recessive. That means, autosomal means it doesn't occur on the sex chromosomes, which we'll talk about next week. And it's harmless, but what it means is that it has abnormal transport of this amino acid. Certain compounds are present, and basically the baby pees blue. It's weird, but absolutely not harmful. So, two parents that have never had blue urine have a child with blue diaper. Okay. If the blue diaper syndrome is recessive, let's give it big B, little b, okay? Then you have to think about, if the parents don't express it, they must have at least one copy of the dominant each, okay? So, dad, well, let's say the child, little b. We know this because they have the recessive symptom. If they express it, they have two copies for autosomal recessive. Okay? But that means that each parent had to each give a recessive gene to them. Because each one of these comes from the mom and one comes from the dad, okay? Let me just highlight these to show you that this one's from dad. And this is from mom. Okay? Let's make this one red so that you know that this is mom, okay? What does the mom have? She has to have at least one copy of the dominant to express dominant, which is no blue diaper. But she has to have given the child this little b. So that must be her. And now we have dad. Dad has at least one big B because he doesn't have blue diaper. But he gave this blue little b to the child, so he must have it. Therefore, he has to be little b, little b. What are the chances that their child has BDS? Well, let's copy paste this, okay? All right. There. The mother, the father. And remember, this is like the cross we already saw, okay? We do big B, little b, big B, little b, little b, little b. All right, next. What are the chances that their second child will have BDS? Remember, it does not at all rely on the fact that they've already had a child with it. These are independent events, independent fertilizations, okay? So let's look at this. 25% chance that their child will be homozygous dominant. 50% chance their child will be a carrier. 25% chance that their child will be little b, little b. So the chance their second child will have it is little b, little b. Don't get stressed out about this and concept here, okay? A lot of people tell me, well, if it's a 25% chance and it was a 25% chance before, then the chance of them having 2 kids with this is 12.5%. Don't stress yourself over this. We're asking about one event. Forgetting the fact that they already have a child with it, their chances have not changed. The chance of their second child, well, every child has a 25% chance. The chance that after the children are born, then we can say the chances that happened post it happening. But when you're actually talking about the chance of it happening in the future for one event, you just talk about that 25%. Okay? All right. I'm going to stop here. Your second recording will be labeled incomplete dominance and codominance. And we'll talk about different ways to apply Mendelian principles.